Today I'll be talking about Crohn's disease in association with plasma antioxidant levels. So a general background, what is Crohn's disease? Crohn's disease is an autoimmune condition, meaning the body is attacking itself, resulting in chronic inflammation of the GI tract. Some symptoms include diarrhea, weight loss, fever, fistulas and fissures in the intestinal tract, and a whole other host of symptoms associated with inflammation of the GI tract. Some pathology of Crohn's disease, there is genetic factors. It's a multifactorial disease, so genetics plays into it with certain populations being higher predisposed, such as Ashkenazi Jews and African Americans. Moreover, if you are a family member who has someone with Crohn's, you have a higher chance of inheriting Crohn's. Moreover, environmental factors seem to contribute to pathology of Crohn's. Stress, smoking, pollution, diet all seem to factor into pathology. The intestinal microbiota is a large part of Crohn's disease, with the autoimmune response being in response to the normal gut flora, which it should not be attacking, but the body is attacking, resulting in inflammation and characteristic of Crohn's. Inherent invasive E. coli strain has also been implicated in Crohn's. And of particular interest to my study was oxidative stress, and that is reactive oxidative species destroying the lipids in phospholipid membranes, resulting in cell death and injury, which promotes inflammation, increases vulnerability to damage in the GI tract, and reduces regenerative ability in the GI tract. So the goal / pathology of this study was, I want to see how oxidative stress contributes to Crohn's disease, and I also want to see, is there a correlation between antioxidant plasma levels and Crohn's severity? This is through a GSA assay, a FRAP assay, an Allen's bilirubin plasma levels, and they also measured Crohn's disease activity index, which was how they gauge severity of Crohn's disease via taking laboratory tests, patient input, and other clinical tests. They also looked at biochemical markers of inflammation, and they did a statistical analysis in 55 patients of varying severity of Crohn's, as well as these healthy controls in order to compare that. So in this first figure, you can see that active Crohn's disease patients have lower levels of plasma antioxidants, bilirubin and albumin, as well as they have higher pro-inflammatory associated proteins, leukocytes, platelets, and CRP, or C-reactive proteins. And they also have lower levels of red blood cells and hemoglobin, which is associated with inflammation. This is a comparison to the inactive Crohn's disease patients who are in remission and the normal PO controls who don't have the Crohn's disease. Moreover, you can see from the FRAP assay, the ferritin reducing ability of plasma, the GSH assay, the glutamine reducing ability, that active Crohn's disease patients had lower levels of these plasma antioxidants than the inactive. So using statistics, they were able to determine correlations between these plasma antioxidant levels in Crohn's disease patients and Crohn's disease severity. As you can see, there's a negative correlation for all of these antioxidant species, meaning that as the plasma levels decrease, Crohn's disease pathology increases, and conversely the same is true. Therefore, active Crohn's disease patients have lower plasma levels, antioxidant levels, and activity. Crohn's disease severity and plasma antioxidants have a negative correlation, and higher reactive oxidative species are connected to Crohn's severity. Antioxidant plasmas could be useful as a diagnostic tool of severity, so they could use that to gauge if a patient might worsen via their high levels of ROSs or low levels of plasma antioxidants, and they can try to treat them preemptively, harsher medications to try to delay the onset of the severity. More so, antioxidant therapies might prove essential in combating Crohn's disease, and in conjunction with normal like anti-inflammatories, immunosuppressants, and antibiotics, they may help to achieve higher rates of remission in Crohn's disease.